Hi everyone, my name is Adhira Ganesh. I'm an 11th grade student from Newfoundland, Labrador, Canada. And today I'm going to be talking about my project, which compared firing rates of neurons from the primary motor and premotor cortex in a macaque monkey. So millions around the world suffer from a variety of different neuromuscular disorders, and these disorders can make it really hard for people to communicate and move on a daily basis. So my project focuses on a technology which can translate your thoughts into actions. Like if I wanted to move my computer cursor from one side of the screen to another, I just have to think that and the hardware will allow the cursor to move. So this technology is called a brain-computer interface, or BCI. So BCIs can do this by harnessing brain activity and translating it into what the user wants, like movement. These BCIs, however, can be very inaccurate. So if I did want to move my cursor from the left side of the screen to the right side, the BCI would not be able to make that movement totally accurate or very precise. So though it can move from the left side to the right side, it wouldn't be exactly where I want it. So my project is all about trying to increase this accuracy. The inaccuracy of BCI stems from the fact that it's very hard to interpret brain activity, and the BCI doesn't know exactly what you want. It just knows like a rough idea. So the portion of the BCI that's responsible for translating brain activity into user intention is called a decoder, and the decoder decodes neuron firing rates, which is also called the brain activity. And basically from that, from those firing rates, it finds out what direction the user wants to move in. So the neuron activity in your brain reflects what you want to do. So if you wanted to move your arm straight, the neuron activity in your brain would reflect that. So to achieve these very precise prosthetics, we need to decode this neuron activity in your brain very well. So there are many different ways to improve the accuracy of decoders. One way is to record brain activity from a brain region which encodes direction well. See, when I move, there are some brain regions that are lighting up and neurons are firing, but there are some brain regions that are not that active in movement. And we want to find the brain regions that are the most active because those neurons will give us the most information that is useful to me. It's almost like if I gave you two maps, one to California and one to Texas, but you wanted to go to California, the Texas map is pretty much useless to you. So just like that, I want to find information that is relevant to me. So this is a hemisphere of the brain, and highlighted in purple is the premotor cortex, or PMD, and highlighted in blue is the primary motor cortex, or M1. And these were the two brain regions I compared in my study. So I wanted to see which brain region had neurons that are more directionally tuned or would give me the most relevant information. So all the data I used in this project came from an open source website. This website, basically it has all the brain regions listed, and when you click on one, it shows you different experiments that have been done targeting that brain region. So I looked at two experiments, one focused on neurons from M1 and one from PMD. The data that was collected from these two experiments were from an adult healthy macaque monkey. And this macaque monkey was trained to perform different tasks while it was moving. And while it was performing these tasks, the brain activity was recorded using an electrode array that was put on the surface of the brain. And the data was collected from a total of 143 neurons. 78 of these neurons were from PMD and 65 were from M1. So right here, you can see this figure. It shows a macaque monkey. The blue dots arranged around the circle show the different directions the monkey was supposed to move in. So they're arranged from 0 degrees all the way to 315 degrees. So the monkey performed different reaching tasks in these directions. And while it was performing these tasks, the brain activity was recorded using that electrode array that was on its the surface of its brain. So the circle here in the brain represents a neuron, and there's a dotted line going to 180 degrees, and then there's another one going to 0 degrees. So this one is longer because 180 degrees was the preferred direction, and this one is shorter because this was not the preferred direction of this specific neuron. So that's kind of stems from the fact that neurons have a preferred direction. So when I move my arm 90 degrees, there will be a neuron or a group of neurons that love when I move in that direction because it's their preferred direction. So when I start moving that way, they just start spiking and firing, and they exhibit a lot of action potentials and activity during that time while I'm moving. So just like that, each neuron has a direction they least prefer. So that direction is called the null direction. So the firing rate in that direction will be the least. So for this specific neuron, 180 was its preferred direction because you can see the vector is longer, and this is its null direction. So it's the direction right opposite. So the firing rate, so when the macaque monkey was moving in this direction, this neuron was firing the most. When it was moving in this direction, it was firing the least. My hypothesis for this project was that M1 neurons were going to be more directionally tuned, or they were going to provide me with more information that is relevant to me. So M1 would be the California map in that map analogy I gave earlier. I formed this hypothesis after doing some research into what brain regions are involved with motor tasks. I found that M1 neurons exhibit more activity during movement, and PMD neurons exhibit more activity right before movement, so while you're still planning on how you're going to move. So that's why I hypothesized that M1 neurons would be more directionally tuned. However, the reason that it wasn't defined or definite that M1 neurons were, like, the fact that this was still a hypothesis was just because scientists don't exactly know what these two brain regions do. It's just like a theory. It's just like M1 just happens to show this, but not all the time. So to find out which brain region provided the most useful data, I calculated something called the Directional Selectivity Index, or DSI. So DSI is a measure of how directionally tuned a neuron or group of neurons is, or how well they respond to movement stimuli. So this will help me determine if M1 or PMD neurons are more involved with movement. So this is the formula I used to calculate the DSI. So here you can see that there's the preferred firing rate minus the null firing rate. So the null firing rate would have been the shorter dotted line I showed earlier. And that's all divided by the preferred firing rate, which is the highest firing rate, plus the null firing rate. And the sample size I used was 62 neurons. 31 of those were from M1 and 31 from PMD. So these are some sample calculations. You can see there's a PMD table and there's the M1 table. And this is just for 4 neurons, but I calculated this DSI for 31 neurons for both PMD and M1. And these numbers were all, they all look similar to that. So these are 2 raster plots that were generated by the code in MATLAB I used. And so on the left, we have a randomly selected neuron from M1. And on the right, there's a randomly selected neuron from PMD. So this one is neuron 122, and this one is neuron 44. So you can see here in the M1 neuron, there's a lot more like blue ticks. And that's just, these blue ticks represent every time a specific neuron spiked. So for this plot right here, neuron number 122, it spiked this many times for the trial number over this many seconds. So you see directions 1 all the way through 8. And here on the x-axis, you can see the time from -1 seconds to 1 second. And obviously you can't have negative time, but the negative is just there to show that it's before the movement occurred. So you can see that there's a lot more blue ticks on the M1 than there is on the PMD graph. And this just shows that the M1 neuron exhibited a lot more activity than the PMD neuron did. So that kind of goes back to my hypothesis that M1 neurons exhibit more action during movement. So to find the difference between the DSI sets, the DSI values I used earlier, I used a t-test. And this t-test was just to find if the difference between these data sets is significant or not. So the alpha value I used was 0.05, meaning any values that were above 0.05, I have to reject my hypothesis. And any values under 0.05 mean the difference between the data sets, it has a very slim chance of being accidental and I can accept my hypothesis. So my p-value actually came out to be 0.03. The exact number is on this figure right here, which means I can accept my hypothesis. And the difference of the data sets, the difference of the data sets that's accidental is about 3%. So the chance that my hypothesis is wrong is about 3%. But I didn't stop there. I went and I further wanted to verify my hypothesis. So I used a decoding model called maximum likelihood estimation, or MLE. So in this model, I feed the algorithm firing rates of a neuron, and it will predict the direction. So the formula for MLE is right here, and it uses probability. So this is the probability of the direction given the firing rate. And N here is just the amount of neurons, which was 143 neurons. So the results I had, the code had all of the firing rates I needed from the experiments that I looked at earlier and imported those data sets into my code. So the code had the firing rates of all 143 neurons in all 8 directions for 80 trials stored in matrices. The input was a firing rate for a neuron, and the output, which was given by MLE, was a predicted direction the monkey was moving in. So the true direction was stored in a matrix, and this was compared to the predicted direction in the MLE code produced to provide a percentage of accuracy. So it was shown that the MLE decoded M1 neurons 12.50% correctly and PMD neurons 5% correctly. So these values are really low because just because of how small the sample size was, but the algorithm did decode M1 neurons better than it did PMD neurons, which further supports my t-test results and my hypothesis. So in the future, to expand on this research, I would like to expand the brain regions I study. There could be a better brain region out there which encodes direction better, better than M1 that I haven't looked at. I would also love to collect data firsthand. This would give me a better understanding of the hardware involved in a process like this. This project was pretty heavy on software, but I would love to see the hardware kind of aspect of that. I would also love to see other DSI metrics. So when I was doing my research, I did find a lot of other DSI formulas. I used one that was accepted and that was very common, except there might be another one out there which provides more accurate results. This is my sample references. So these are just a few of the journals, papers, and websites I looked at for this project. And that's the end of my presentation. Thank you.
Super interesting. One of the questions that was submitted during your presentation was about the preferred neuron directions. Are they similar in both humans and primates, or do you have to sort of remap neuron direction or preferred neuron directions for each species or even each individual?
Yeah, so basically this study did look at monkeys, and neurons in monkeys do behave a little differently, but the basic fundamental concept is still there. Like our neurons would still have a preferred direction when I move. So obviously I couldn't use the macaque monkey data set and expect those results to translate to a human, except the same basic fundamental ideas are still there. So if I were to perform it on a human, go through all the laws and safety first, but if I were to do that, then it would still, the idea that neurons have a preferred firing rate and a no firing rate would still be there. So I could still use that formula.
Got it. Super fascinating.
Thanks
so much, Adhira. Appreciate it.